spermatids, all right? Don't worry about that necessarily, that's more 318 stuff. But what you do need to know is those immature spermatids travel into the epididymis where they mature into actual mature sperm cells. And then the sperm cells will travel through this vas deferens, all right? And it'll loop behind the bladder, all right? So these are going to be the ureters from your kidneys bringing urine to the bladder. These go behind the bladder where these seminal vesicles, remember, add that first fluid to create semen. All right, and then you see that this then travels into the prostate gland where it becomes the ejaculatory duct right here. And so that, remember, that prostate gland is the number 2 gland that adds that milky substance. And then it goes through the Cowper's gland right here. All right, so these 2 little orange balls are going to be the Cowper's gland, and that's what sort of secretes that final alkaline substance before it goes through the urethra. All right, so this is just giving you sort of a spatial context for what we're talking about. The main reason we're showing this is because what you guys need to ID on your exam is just a side view, sort of a sagittal view of this. All right, so if the body is cut down the middle, you can only see what's happening in the middle of the body. But what you can't see is the connection of the vas deferens from the testes. So if you look at the models here, there's nothing until you see this little white noodle-like structure in the prostate gland. What this is doing is helping you connect this all the way to that structure that's in the prostate gland. So when you look at it from the front view like this, you can actually see that the vas deferens loops behind the bladder and then it enters the prostate gland, which is what you see here as the ejaculatory duct. So remember, the vas deferens and the ejaculatory duct are the same structures. We just specifically call it the ejaculatory duct once it's inside of the prostate gland. Are there any questions on the flow or anatomy of any of this before we move on to the female anatomy? Nope. All right, so again, make sure to look at the models up here, right down the middle here, and then you're looking in at that. So you have the ovaries over here, all right, and so that's where you have follicle development, and then you have an ovulation event. So the ovulation event will release an egg into these fallopian tubes. It's labeled oviduct here, but that's what these are. All right, so you have fertilization also occurring in the fallopian tube, but the fertilized egg actually implants in the ovary, all right? And then remember, during pregnancy, the cervix is what dilates, and then the baby is going out through the vaginal tract. All right, so you can see again, the anatomy is fairly simple, and the flow chart is really simple for this as well. So it's just a matter of understanding where everything is located, and then all the different components of the flow chart. Are there any questions over this? We're not going to get too much again into the whole follicle development. Again, that's sort of saved for whoever takes 318. But one thing that I want to mention before we end up moving on, all right, is going to be the actual formation of sperm and eggs, all right, so in terms of meiosis. Does anyone remember that term? Diploid, all right, and so that's being broken down into 2 more diploid cells, all right, so remember you have DNA replication, and then you have the cell splitting into 2 still diploid cells, so that's mitosis. And then what are the end products of meiosis? Right, so you end up doing this. All right, so they still go back to 2N, but then these go through 1 more division, all right, and they make N, which is going to represent a haploid cell, all right? So this is diploid and then haploid, all right? So this is what we're talking about with meiosis. However, with respect to sperm and eggs, all right, so if we start with sperm, all right, you have a cell here that's diploid. So this is going to be, I'm just going to label SG, so that's a spermatogonia cell, all right? So that gets broken into 2 more diploid cells, and then just like the diagram shows over there, that gets broken into these haploid cells, all right? And so these are going to be the early or immature spermatids, and then you'll see over time, so those migrate. Do you guys remember what where they migrate to mature? Right, so these go to the epididymis, all right, and then that's where they mature into sperm cells, so mature sperm cells. All right, and so these are 4 viable sperm cells, all right? So when I say viable, it means that they're able to fertilize an egg, and they're still alive. All right, so we're starting with an egg, all right?
The first one was for the male?
Was that? Yeah, this is sperm. All right, so we're starting with an egg cell, all right? So you're going to have the first division, all right? you have the, we're not going to go through sort of all the mechanics of this again, but what happens is you have the formation of a polar body and then a diploid cell. So this is a polar body. And essentially what that is it's just an inactive haploid cell. So this never goes on to being fertilized. All right. And then you have this developing into more polar bodies. All right. And so just to sort of not confuse you guys, I'm just going to draw it a slightly different way. All right. So there is a diploid intermediate here. And then this goes back into haploid cells. All right, so what happens though right here is with the diploid egg cell, that actually goes through another division of producing a diploid, haploid and polar body. So the main thing I'm trying to show here is that one of these is supposed to be a lot larger than the other. All right, so the polar bodies are going to be really small in size, and then you're going to have the mature haploid egg cell that's going to be a lot larger. And so these three polar bodies here are inactive. All right, so one thing to get out of this is comparing the formation of sperm versus egg cells, the number of viable cells that are produced. All right, so 4 versus 1.